Thank you. It is really, really, really exciting to be here. It's my first time in India, and I'm absolutely loving it. It's just been such a thrilling city to come into, and in Mumbai, and I'm hoping to explore some more tomorrow. And it's really actually very appropriate for me to be speaking after Andrea. She's the work that she presented in showing those sorts of new technologies of imaging and microscopes and how to show the small world is really, it's a tremendously exciting and a really rapidly booming area of research. Where my work comes in is in the gaps between the areas where we can't see things, where we know we have lots of different ways of probing the microscopic world, but we can't actually see everywhere. We can only see very limited amounts due to limitations of technology or because of the physics. And so my work is really there to interpret or to represent what we can't see. So I'm a biologist by background, but for the last, and I specialize in molecular and cell biology. And so actually my undergraduate and postgraduate work was all dealing with microscopes and time-lapse microscopy. But for the last 15, 20 years, I've been working really at, well, at the convergence of two different booming areas. One is biology, which is moving at a great rate, but also in technology with visualizations, computer graphics, which is driven by, well, actually primarily by the gaming industry now, but also in the sheer raw processing power we have with our computers. And really my work is trying to interpret scientific data from biology and to represent it through visual graphics. And my work gets used in museum exhibits, documentaries, and television programs, and lots of different education projects. So what I'm going to be showing you today is a little bit of work from actually my education projects. I'm going to focus on two topics. One is on DNA visualizations, and the other one is on visualizations of malaria, which is a very important disease in India. I'm going to start with DNA and begin with these two model builders, which I very much follow in the tradition of building models with whatever tools you have at hand. This is James Watson and Francis Crick, who built the first model of DNA with these bits of wire and just constructed with cardboard and wire. And the project I'm about to describe, I was working with James Watson for the 50th anniversary of the discovery of the double helix in 2003. So the work I'm about to show you is from 2002 and represents the science of DNA at the time. So they using hard work and data from Rosalind Franklin, who actually did all of the hard science, they then saw this data, but then quickly were able to interpret it and then build a model to represent the DNA helix. This is the DNA helix here. And this is DNA as it's usually shown in textbooks. And it is an accurate model of DNA. It's from X-ray crystallography, and it gives you a sense of the twisting helical structure of this, the atomic structure of DNA. But it really doesn't give you any sense of the nature of DNA. It sort of shows these sort of rigid, shiny spheres and only this little tiny short structure. This is a real image, or an image derived from a microscope, an electron microscope. And with this direct observation technique, with using electron beam to image, this is DNA again. And you start to see that DNA is actually a very long molecule, a very dynamic molecule. But with this direct observation technique, you can't see that helix, that spiraling helix. So really that's what my work does, is bringing many, many different forms of data and little fragments of data and then interpreting it into a way and presenting it to the public so we can actually see what's going on, or if you could see it, this is what it would look like. So I'm going to present some animations now that are from the 2002 project to celebrate the discovery of the double helix. And the animations were designed primarily for classrooms, but then they had a whole life beyond that. And really, I guess the main idea behind my work is not to ever dumb down or simplify the science. The more detail, the more technical accuracy you can put in there, the more interesting they become, the more interesting and the more the public love the science. The problem with science in explaining to the public is the complicated verbal language people use. It's not the ideas, or if you show people the science, they get it and they'll understand. So anyway, off to the animations. So again, here's DNA in its classic double helix form. And if we unwind the double helix and unzip it, you'll start to see a structure. So again, this is from X-ray crystallography, so it's an accurate model. these things that look like teeth here are the letters of genetic code. There's 25,000 genes you've got written in your DNA, or 30,000 genes you've got written in your DNA. This is where this is what they particularly talk about, the genetic code. What I'm going to talk about is more the physical nature of DNA. We'll begin with these two strands, which run physically in opposite directions, which creates a number of complications for your cells, as you're about to see. So these two strands run in opposite directions. The genetic code is written in different directions. Most particularly when DNA is being copied. This is the real main challenge for your cells, is when they're copying the DNA. This is an accurate representation of the actual DNA replication machine you have inside your body. So this, at least the science of 2002, this is what we understood it to be. The DNA is entering this production line from the left-hand side here, and it hits this collection of proteins, what proteins look like, down to the molecular scale. And they're ripping apart the DNA strand and making an exact copy. One strand is being copied directly, and can be seen being copied off to the right-hand side here. But the other strand runs in the opposite direction. This creates a number of complications for your cells. This machine throws out the DNA into these loops and copies the DNA in the opposite direction. Now this representation is actually happening in real time as it's occurring inside your body. I've left out details like error correction. There's a lot of other proteins involved that we knew were involved, but I left out of this visualization to make it observable to a human. But this is a reasonable representation, at least in 2002 science, of so you have billions of this actual machine right now working away inside you at this speed, copying your DNA. This is really the nature of molecular machines inside of our bodies. And the DNA animations I'm about to show you are all more of these sorts of macromolecular machines that get together and how they operate. And I hope, I know we have a lot of engineers, so people are interested in engineering and also in chemistry and physics. What I hope you realize is that biology is full of these sorts of molecular, the molecular engineering. The same sorts of problems and issues we have in our big macro world are occurring and are solved by our cells inside our body. So what I'm about to show next is really updated science. It's showing you, this was done in about 2010, it's showing you the current state of science for DNA science at the time, but also updated technology, because computer graphics have just really transformed over the last 8 or 10 years. When I built this particular visualization, I had a $50,000 US dollar computer and a $20,000 software, and it took me, I don't know, months to actually render and produce that visualization. Nowadays, with this laptop and with free software, I can easily do much better. So essentially, if you have access to a computer and access to the internet, you can start building these visualizations of your data and representing them. It's really quite a transformative, and just opens access to creating these sorts of visualizations to anyone. So again, we begin with DNA. This visualization doesn't have sound as yet, but it's coming. So DNA is really quite tiny. It's about 2 nanometers across, about 20 to 30 atoms across. But in each one of your cells, a typical strand of DNA is 30 to 40 million nanometers long. So 2 nanometers across, but 30 to 40 million nanometers long. So to keep the DNA organized and to regulate access to the genetic code, it's wrapped around these purple proteins, these proteins called histones here. All of this field of view is still that single strand of DNA. And this huge package of DNA is called a chromosome. And we'll come back to chromosomes in a minute. This is a representation of the chromosome and of various other proteins. We're pulling out, zooming out through a nuclear pore. This nuclear pore is like a gateway to this compartment called the nucleus that holds all the DNA. We're pulling out, zooming away from, these are all the different structures inside the cell. And we're pulling back to a view that if you look down a typical light microscope is a living cell. And in fact, this is a real footage of a living cell. This is the nucleus, so that compartment that holds all the DNA, and this is that living cell filmed over time lapse, so that's why you can see it moving. So the nuclear envelope breaks down. These purple sausage-shaped things are those chromosomes, those packages of DNA, and they go through this very striking motion. They jiggle, they wiggle, and move back and forth. And then when the cell feels that everything is aligned and ready to go, it rips apart the chromosomes. One set of DNA goes to one side, the other side gets the other set of DNA, identical copies of DNA, and then the cell splits down the middle. Now again, this is under time lapse. This happens in about 2 to 3 hours in real time inside our bodies. Now we're going to rewind and we're going to revisit the chromosome and look at another visualization of its structure. So here we are, again, rewound. This is the equator moment where the chromosomes have lined up. And we're just going to isolate just one chromosome, one package of DNA, and pull it out and have a look at the structure. So here's that one chromosome. These things that are sticking out either side of these whiskers are part of the dynamic scaffolding of the cell that are constructed and deconstructed very rapidly. And they're used for orienting and organizing the location of things inside the cell. And this is the chromosome, these two sausages. One strand of DNA is bundled up into one sausage. The other strand of DNA, the identical copy of DNA, is in the other sausage. What we're going to focus on is this red region, which is one of the biggest macromolecular structures we've discovered so far inside our bodies. It's the interface between the scaffolding, the microtubules, and the actual chromosome. It has multiple roles, really fascinating roles. It's very much involved with holding onto the microtubules. It's also involved with growing and shrinking the microtubules, coordinating the movement of the chromosome. It's red here because it's also a signal broadcasting system. It broadcasts through chemical signals its state of readiness. Because what it's also able to do is feel through mechanical tension whether the chromosomes align and everything's ready to go. So it's made up of about 200 different types of proteins, thousands of proteins to get in total, that all build this huge molecular biophysical machine. Now it's turning green here because it's feeling through mechanical tension that everything is just right. But one little last bit is remaining red, and that's that signal broadcasting system, which broadcasts a stop signal. And it's literally walked away down the microtubule when it feels everything's ready to go. So one last animation. These are little molecular motors, little courier molecules. The orange ones are called kinesin, and they can walk one way. And this is dynein walking away from that kinetochore with that red broadcasting system on its back. And they've got these long legs. So they take little 16 or 13 nanometer steps, where kinesins can have little short legs, and they can only take little 8 nanometer steps. And what's also interesting is that there's a direction to these microtubules, this highway, and the kinesins can only walk in one direction, and the dynein can only walk in another. But again, these are simulations based on representations based on many, many different forms of data that are brought together and represented and shown that we don't have any other way to see. There's probably 200 to 300 papers went into just this visualization alone. Many little fragments of data, because typically in research of this kind of sort, there are labs that focus on individual proteins and what those proteins are associated with. But it's very difficult to bring those forms of data and look at the whole big picture. And that's the nature of the sort of work that I'm doing. There's a tremendous need for this kind of work, and particularly with all the different forms of science that are absolutely booming. There is just a huge amount of data coming in, and there's a huge, tremendous need to show the public, to explain what is going on, to inform politicians, and also, particularly, to explain to students what the nature of this work is about. So with my remaining time, I'm going to now switch to malaria, which is a tremendous burden. It's one of the most important economically, but also as a disease, a burden on the world. I'm going to show you a visualization of the nature of what malaria is about, but also I'll show you some of the forms of data that go into these sorts of visualizations. In India, there's about 1 million, or more than 1 million people every year who are struck down with malaria, and many, many famous people have been debilitated by it. Tens of thousands of people die of it every year within India. And but if you have a million people, each one of those has a family that if the member is sick, the family members have to stay home and take care of them. And particularly when you're a farmer or life is more difficult, if you have to stay home and take care of the person who's sick, or if you have a person who's going to go out to work, it creates a tremendous economic burden. Not only is just the suffering that the disease causes, but it's also the economic burden on the region. So that's kind of one of the motivations for why we do this kind of research, and to try and solve and alleviate the world from these sorts of diseases. So just to give you a quick overview of malaria, this is one of the best diagrams I've ever found of the malaria life cycle. It's very, very complicated. That's one of the big challenges I've had, is to represent the science accurately, but it is a complicated disease. It's a very devious parasite and how it infects our bodies. What we're going to focus on, so this is the mosquito biting the skin. This is a representation of that. The parasite enters the bloodstream and then gets to the liver, into into your liver within a minute or two, and then it infects the liver, bursts out, and then this is describing the cycle inside the blood cells, and that's what actually causes the symptoms that we see, the disease. But then it gets back into mosquito through a different stage, and then there's a whole other life cycle. The mosquito actually is infected by malaria as much as we are, diseased by malaria as much as we are, and it's not the mosquito's fault. It's really just the parasite and the way it swaps and has different tactics for dealing with different organisms. So I'm just going to describe the human side of the life cycle, but there's a whole other animation that describes the mosquito side. This is the forms of data that go into it. This is very simple forms of data. This is an electron micrograph of a mosquito's head, and the sorts of information I take. Here's the proboscis, the mouth parts of the mosquito, and it's kind of covered by these feathery parts and the antenna and all the different structures. All these sorts of little details I'll bring into the animation. This is the biting stance, or the way the proboscis works when it bites a human, and it has a sort of a sheath that bends back called the labrum. And then what I found interesting is the proboscis is very flexible. It's actually got a bunch of blades that chop up the skin and pierce the bloodstream, but also are very flexible, so it can bend in and sort of suck up the blood. This is, again, another type of electron micrograph of blood vessels just underneath the skin. And what these folds here are actually of a fingerprint, the folds of a fingerprint. So these are the capillaries and other blood vessels. Very much these sorts of details are informative for the animation. And this is some footage from Japan of a surgery on the liver. Of course, I get informed by the textures and the structure and so on of what the liver looks like. What I also found interesting is the heart just up here beating away is really causing the whole liver to move and to bump. And the movement and the dynamic nature of these sorts of footage very much inform the animation as well. Now it's a bit hard to see here, but this is a bloodstream. Actually, these are little beads that are they've inserted in the bloodstream. They're attached to a type of immune cell, a white blood cell that sits as resident in the liver called a Kupffer cell. And those are the cells that the parasite targets. So again, these sorts of images you'll see hopefully reflected in the animations that really very much inform how the animations come out and how they look. And one visualization, this is time-lapse footage. These are red blood cells that are lying on the slide, and the parasite is really, really tiny. And I think it's this one here. Yeah, it's this one here. That little sphere there is actually, they're kind of more light bulb shaped. But this is the actual mode of entry into the blood cell. It attaches very much sticky. It's very sticky for red blood cells, and it reorients itself, and it really shoves its way in. It's quite a rough process. And there it goes into the red blood cell and takes over the control of that red blood cell and digests the contents. So again, footage that very much informed the visualization. So this, I've done my own narration on this one, so I'll let it play. It runs for 3 minutes, and then I'll be finished. So it gives you an overview. It's very much aimed at education to show the total nature of the parasite's life cycle. I'll go ahead and play.
The malaria parasite is an ancient organism. It has been with us since before we were human. Famous victims include Alexander the Great, Genghis Khan, and George Washington.
And Gandhi had malaria too.
The malaria life cycle follows a devious path, swapping back and forth between mosquitoes and humans. This mosquito is infected with the malaria parasite. Because she is pregnant, she has become hungry for human blood. During the bite, she injects saliva to stop the blood from clotting. Her infected saliva also carries the malaria parasite. The parasite rides the bloodstream like a network of roads, seeking its first target. The core of your body's blood filter system, the liver. Sensing its arrival at the liver, the parasite searches for an exit. A sentinel Kupfer cell is the entry point to liver tissue. Leaving the blood, the parasite infects a liver cell, killing one or more other cells on its way. Over the next few days, the parasite undergoes hundreds of nuclear divisions, copying its DNA over and over again. A single infected liver cell can create thousands of new parasites. The next generation of parasites are modified to infect a new target: red blood cells. Inside a red blood cell, the parasite can hide from the body's immune system. The parasite slowly devours the contents of the infected cell and creates more parasites. The infected cell becomes sticky and grips onto blood vessel walls. Once mature, the infected cell bursts, spreading more parasites through the bloodstream. Malaria victims suffer fever, loss of blood, convulsions, brain damage, and coma. This year, 10% of people on Earth will be struck down with malaria. Countless millions have been killed by it. Most people who die from the disease are pregnant women and children under the age of 5.
In the narration, the 10% statistic is not, it was accurate at the time, or there was an estimation at the time. It was revised down to 4%, and now it's back up to guessing about 8%. It's very hard to actually get accurate numbers on it. But there's still the economic burden, particularly. Because, I mean, we do have millions of people being infected every year. Really, what it does to a region's economy is tremendous. So, this is kind of the reason why science, particularly medical researchers, are doing the research, is to eradicate these sorts of diseases and to really improve the world. But really, I hope you saw in the molecular stuff, the DNA visualizations, the nature of molecular science is completely mind-blowing. It's just so exciting and extraordinary to be out at the frontier of human understanding and to be discovering all this new information. But really, there's lots of very important motivations, such as to eradicate diseases like malaria or cancer or diabetes, which really motivate most medical researchers. So yeah, I think I'll stop there. So thank you.